Yeah. So basically the project is OpenMDL, and basically what we did during our lab, in our lab, is that we use MD simulations to analyze the protein ligand interactions, and we have mainly a lot of pharmacists, so that basically they then prefer to have like software that is quite easy and user-friendly to use. And we previously used quite often Desmond, but the idea was that we basically then switch to something else, which would be then more open source and user-friendly. And this is how then the idea was that we switch from Desmond and the MD simulations in Desmond to OpenMM and then MD simulations in OpenMM. And I got then the task to basically prepare a workflow that would allow every user to basically set up the MD simulations of his protein and ligands, run the MD simulations, and then afterwards more or less analyze the protein ligand interactions that are happening during their trajectories. Yeah, basically this is how then OpenMDL happened. And OpenMDL is basically here, as the name says, is then simplifying the complex. It does the building, simulating, and analyzing the protein ligand systems in OpenMM. Yeah, and basically what it is that it's a workflow for MD simulations of protein ligand complexes, and it consists out of 3 parts, those being the setup, the simulation, and the analysis, so that every part of them is responsible for the different parts of the workflow. And here's then also the QR code on the right upper part for the GitHub page, where basically you can then see the tool. And basically here's then also a little bit more detail of what each of those parts does. The setup is basically done to prepare the MD simulations, so that basically you have then an interactive web interface that allows you to prepare your MD simulations, so that you use your files to prepare them. Then the simulation is responsible for the backend, the preparation, simulation, and post-processing of the simulation. And the analysis has then different paths to analyze the protein ligand interactions and the trajectories, those being the binding state calculation, the interaction analysis, and the water Wilson calculations. And what I would do in this presentation is basically that I go one by one to all of the parts of this workflow to explain more in detail what they do. And we start off with the setup. And the difficulty is basically quite often with the setting up of the MD simulations is that you have quite a lot of different variables that can be complicated, especially for people who basically are new into MD simulations. And the idea that we had was that we basically create, first of all, a setup, which is basically then a web interface that is more user-friendly, which allows then an interactive preparation of your simulation files and allows a flexible modification, so that if you want to modify different variables, for example, the non-bounded cutoff, you can modify them, but there are directly already default settings in place, so that people who are then less familiar with it can basically directly just run it out of the box. They just need to say what protein they want to simulate with what ligand. And here's basically a more detailed overview how it looks like. It is basically based upon OpenMM setup, the OpenMDL setup, and it has 2 different options of the preparation of the files, those being the PDB fixer preparation and the Ember tools preparation. What the user gets is that basically he can then select if he wants to use the receptor and ligand with the PDB and SDF files, or if he then directly goes to the Ember tools and basically modifies them with the Ember tools. So there's the option that if you have PDB files, you prepare them with PDB fixer, or if you have PDB files, you can convert them to the Ember files, which is directly also implemented in the OpenMMDL setup, and basically then directly afterwards have all of the necessary settings and necessary files for your simulation. And basically how it works is that you have a command line and you run OpenMDL setup. And with that, you get in your preferred browser the page that opens up OpenMDL setup, because it's done with Flask. So basically what we have here are HTMLs that open up one by one, and you click to them to prepare your MD simulations files. And basically here's how a page would look like on the right side, that you would see that the user would have the option to select the PDB and Ember, and you would basically go then one by one to the pages, which would basically then allow and explain here how to prepare the MD simulation files and what the necessary settings are. And in the end, basically, it would be that he can select then the chains that he wants to simulate, the force fields, and the membrane and water models, how big the water box would be. So all of these can be then modified and adjusted. And here's also how the last page would look like, that you would be able to modify the length of your MD simulation, the equilibration length, and other important simulation settings. And in the end, you would be able to directly then save over the files that you prepared, and you would have then 3 output files. Those being, in this case, the PDB file for the simulation, the SDF file with your ligand, and the script that you would then directly use afterwards for running the MD simulation and OpenMDL simulation. So that the setup is mainly used for basically doing an interactive preparation of the MD simulation files. So that is then a little bit easier for the user to see what he needs to select, how to prepare the MD simulations. And when he has them, he can directly use the files that are the output of the setup directly in the simulation. And the simulation is then more or less the backend of the simulation in OpenMDL. And what it does is basically that it does then it takes your ligand, your protein, and a cofactor, if you have one, and puts them all together, then solvates them or as a membrane so that the system is then prepared. Then it runs the MD simulation, and afterwards, it's then quite often the case that after you have the MD simulation, there are some annoying things like your protein is outside of the water box, or the water box is then moved from your original coordinates. And what we directly do is basically that we then do the post-processing. For this, we then use MD-Trash and MD analysis. And what we do in the post-processing is that we put your protein and ligand back into the water box and move it back into your original coordinates. So that in the end, you have the topology files, the trajectory files of your simulation, and you can directly then use it for the analysis part, so that you don't need to modify them or somehow post-process them after the OpenMDL simulation. And those can be then directly used for the analysis part, with the analysis part consisting of different tools that help you to analyze your protein and ligand interactions during your MD simulation. And basically, this is then the first part that we would have, the stable water clustering. And what we do here is basically that we look at the waters and look at the movement of them. And then we analyze how far the water molecules move during the MD simulation. And then we can put a threshold upon them, how far they moved, and to that create clusters, which are then the stable water clusters, in the sense that basically we see that, for example, this water moved not so much during the MD simulation, so that is quite a stable water, which we can then directly use afterwards for drug discovery or drug optimization, to either using this water molecule for water bridge interactions or basically then somehow replacing it. So this would be then helpful. Then the second part that we have is then the binding mode generation, which is basically that we then have the trajectory of the protein and ligand simulation, and we go frame by frame and look at the interactions that we have there. So that currently we use for this PLIP, which is then the protein ligand interaction profiler, which was created by the TU Dresden. And it is basically either a web server that you can then directly access and look at your protein ligand interactions, or the Python package, which is then the package that we use in OpenMDL. So that the Python package directly gives you the same output as you see here on this page. Basically, that you see what residue does interaction with what atom of the ligand and what the distance is and what the interaction is. This would be the first option that we currently use. The second option that we want to implement in the next release would be the usage of PROLIF, which is also another tool called protein ligand interaction fingerprints. And it basically does more or less the same as PLIP, so that it looks at your frame and looks at the protein ligand interactions and then says if there is an interaction present or not and what interaction it is. And the reason why we first went with PLIP and not PROLIF was mainly the water bridge interaction implementation, because PROLIF at that time didn't have a water bridge interaction implementation. Currently, with the newest release that should be appearing soonish, like in 1 month, water bridge interaction would be implemented, and then it would be then reasonable to implement it in OpenMDL to have then PROLIF, because it now has all of the necessary protein ligand interactions, because previously it didn't have the water bridge interactions, but PLIP did, so we used PLIP. And basically, to quickly also explain what PROLIF is and how it works, is basically that it's a package that is based upon MD analysis. And MD analysis overall is a package used for analysis of MD simulations and other stuff. And it's part of the Google Summer of Code, which is basically like a project that allows different researchers and different people to participate and contribute to open source packages, where basically MD analysis and PROLIF in it are part of this organization that is then sponsored by Google Summer of Code. And Google Summer of Code basically then allows different people from different backgrounds to participate and contribute to MD analysis. Basically, what it does is that Google Summer of Code gives money to some of the contributors who then contribute to MD analysis, and they basically contribute to different projects. And here are two projects that I would quickly highlight because I then also mentor them. Those being the projects from Stuart Young and Neil Wimmer, and they are basically both ProLift connected, so that both of these projects contribute to ProLift. And the first one from Stuart is the H-bond interactions from implicit hydrogens. And the second from Neil is then the enhancement of the ProLift visualization, so that you can see on the right lower part how the visualization looks currently, and the idea is to optimize it so that it's better from the visualization point. And these are basically the projects that I currently mentor with Cedric, who is then the main creator and maintainer of ProLift. And here, basically, as soon as we have then the protein ligand interactions, we can then create the binding modes. Because what we have here is basically for blip, it gives us all of the necessary information that we need. And we can then create this protein ligand interaction fingerprints for the binding mode so that we create these combinations of interactions that you can see down below with zeros and ones. And basically the idea is that for each of the interaction, we look at the combination of the interaction type, the protein residue, and the ligand atom. So you can see on the right upper part, for example, one example would be that isoleucine 403 does a hydrophobic interaction with these atoms of the ligand. Or for example, we have glycine 351. It acts as a hydrogen bond donor towards the ligand atom 24458, where basically the ligand is then the hydrogen bond acceptor and the glycine is then the hydrogen bond donor. And we can basically, with these combinations, then create these fingerprints that you can see down below, with an interaction being present being 1, and if it's not present, it being a 0. For example, binding mode X, we have two phenyl rings. Both of them are acting as hydrophobic interactions with a specific residue. And if there is an interaction present, then we give it as 1. If an interaction is not present, for example, like here in the triazole, then we give it a 0. Whereas as you can see in the binding mode epsilon, we see that the phenyl ring, one of them loses the hydrophobic interaction, so that basically is then not 1 but 0. But the triazole ring gets an interaction and it's then a 1. So that basically we can then say that the combination 1, 0, 1, 1 is a different combination than 0, 1, 1, 1. And basically we can then look at the combination of interactions between the ligand and the protein present in a certain frame. And we then calculate these fingerprints, the combination of interactions, and we check how often this combination of interactions appears during the MD simulation. And here, for example, we see that the binding mode X appeared in 15.3% of the frames that we analyze, while binding mode Y appeared in 3.8% of the MD simulation frames, so that we see how often those appear during the MD simulation. And this allows us also to then look at the Markov state transition graphs, which basically the idea is that if you have, for example, binding mode X, and you have these combination of interactions in the beginning of your MD simulation, you then run your MD simulation, and at some point your ligand moves to another part of the protein, loses those interactions, and gets the other ones. For example, it then becomes binding mode epsilon, Y. You then see that there's a transition from binding mode X, which was in the beginning of the MD simulation, to binding mode Y. So that the Markov state transition graphs basically help you to see the transition from one combination of interactions to the other one, and if basically it's reversible or not. For example, if binding mode X moves and goes to binding mode Y, if it can go back, if the ligand goes back again to the previous combination of interactions, or if it then stays there and is still binding mode Y. So that it basically then helps you analyze the movement of your ligand in the MD simulation. And this is then the idea to analyze a combination of interactions, but it's then also quite often the case that sometimes you need to look at a certain interaction so that you don't need the combination of interactions, but you want to have like this specific hydrogen bond acceptor interaction. And for that, we then also have the interaction bar plus implemented. We basically just use them and see for a certain interaction how often it appears in the MD simulation. That you see that here, for example, the interacting atoms A and B, this interaction between them appears at 19.88% of the MD simulation. So this is just an example. Usually, basically, you then also have the dimension of what ligand atom it is, with what residue it interacts, and what the interaction type is. And to make it not just 2D and not barcodes, but also 3D, we then directly use NGO to visualize it, because we then also have in blip the ligand coordinate atoms of each of the interactions that happens. So basically, for example, if we want to see a specific hydrogen bond accept interaction, we have the ligand coordinates for each frame where this interaction happened. So we can just use these coordinates to showcase how the ligand moved, if it was stable during this simulation, direct interactions, or if it somehow moved. So that, for example, here you see the yellow and red clouds, and the yellow clouds then showcase the hydrophobic interactions, and the red ones show then the hydrogen bond acceptor interactions. And we have then in the NGO and Jupyter notebooks the option to basically select what specific clouds you want to see, what specific ligand interaction you want to see. That, for example, if you have a preferred interaction that you want to see, you can then see how close to each other the clouds are. And for the water bridge interactions, we then obviously have also the water bridge pie charts, because it's quite often for the water bridge interactions that they either happen so that one water molecule does the interaction, or it can be the case that the water molecules move, the other water molecule appear, and that basically different waters participate in the water bridge interaction, so that one water molecule replaces the other one in the interaction. And here we basically see how often the specific water ID, the specific water molecule, participated in the water bridge interaction. So for example, here we see that the water ID X participate in 50% of the interactions in this specific water bridge interaction, but then it moved away and we had water ID Y and water ID Z replace it, with one doing an interaction in 29% of the frames and the other one in 22.5% of the interactions. And here then also a couple of examples that we used in the paper to basically showcase how it can be used during different examples and different case studies. And the first one was done by a colleague of mine, Leon. He used the stable water clustering, and he was then also one of the main coders of that part. And basically what he wanted to see was for the ALK2 kinase that there was a specific water bridge interaction that was important. And he basically here validated the stable water clustering and the water bridge interaction analysis. And here you see on the left lower part that there are two ligands, and they both interact with the ALK2 kinase. And one of them was basically more potent than the other one. And the reason behind it was because of the water bridge interactions that happened. That you can see in the middle picture that there are interactions happening with the lysine, with the glutamate and aspartate, whereas on the right figure, there is only an interaction with the aspartate and glutamate. So that we could see that the reason was mainly that we had these water clusters on the left part, and the waters in them interacted for one of the more potent compounds with all three of them, whereas for the other one, it interacted only with two residues. And we also saw that for the middle one, the more potent compound, the interaction frequency was also higher, so that it was here around 26% of the frames of the MD simulation, whereas for the other one, it was only 18%. The other part that we analyzed was then the binding modes. And here, in this case, I was the one preparing it and validating it. I use, in this case, the tolerase 8 homodimer. And basically, this is how a homodimer looks like of the tolerase 8. And in the middle part, in the interface between both of these between the interface of these homodimers, there's a binding site where a compound is located. And what we did was basically we looked at the interactions of the ligand with the protein during the MD simulations to see how the binding modes are happening and if there's a transition from one binding mode to the other during the MD simulation. And what we saw was that there were three dominant binding modes happening, those being binding mode 2, 3, and 4. And we could also basically then see the transition from one binding mode to the other. Here then also the 2D structures displayed how it looks like that we had in the beginning binding mode 2, and binding mode 2 had hydrophobic interactions of the alkyl chain and the phenyl ring. And what happened then during the MD simulation was that the ligand moved a little bit. So we then saw that it lost the alkyl chain hydrophobic interaction, and it became then binding mode 4, where basically we then saw that the phenyl ring still does the hydrophobic interactions with tyrosine and phenylalanine, but the interaction between the alkyl chain and the tyrosine is now lost. And after we analyzed the MD simulation further, we then also saw that there was another movement, and it then became the binding mode 3, so that the ligand moved a little bit further, and then an additional interaction appeared, now the hydrophobic interaction between the phenyl ring and the valine. So that we then saw that for the binding modes in the beginning, it was one binding mode, then the ligand moved, it got to another binding mode, and then it moved further and got to the third binding mode, and we were then able to see during the MD simulation how this transition happened from one binding mode to the other. And finally, for the final validation, or one of the other case studies that we had, we wanted to analyze the metal interaction, and in this case we then had the human aromatase SUB19A1, and there we looked at letrozole and the interaction of letrozole with the heme. In this case, for the heme, since it's a cofactor, we then use the amber-tooth preparation, because the PDB fixer preparation mainly is used for protein and ligand MD simulation, so cofactors is there a little bit more trickier, so that we use then the amber-tooth for the preparation. And we then in this case focused on the metal interaction, and as you can see, we then saw that it had 46.22% of the frames, the interaction between the letrozole and heme, showing that it was quite a stable interaction, at least in some parts of the MD simulation. And yeah, basically here's then also quickly I highlight how to use OpenMMDL. And basically the idea is, or the usual standard workflow is that you have the setup that allows you the preparation of the MD simulation files, then you have the OpenMMDL simulation, where you run your simulation, and finally you have the analysis, where you basically analyze your MD simulation and the protein ligand interactions. But the good thing is that basically all of these three parts are kind of connected, but also not connected, in the sense that, for example, you can use the setup to prepare your MD simulation files, use simulation to run your MD simulation, but then for the analysis, don't use analysis and use another tool that you want. For example, one would be the MDPath that another colleague of mine created. And basically the idea is that you can look at the allosteric paths and the activation paths of the GPCRs. At least this is what he then did in this tool that we created. And the good thing is that you can directly then use the output and the prepared MD simulation to use it for this analysis or any other analysis of your trajectory that you want. Or you can directly use already the MD simulations that you have somewhere and directly use them as input for the analysis, so that you don't need to always run a new MD simulation to analyze it. If you already have some other MD simulations from some other engines that you curated and just want to use the analysis parts of an OpenMMDL, you can also directly use the MD simulations that you have as an input and basically just use then the analysis tools of OpenMMDL to analyze them. And here's then also quick again a wrap up what was done and what the separate paths are for, with the setup being a step-by-step selection of the simulation settings. It has two paths for the preparation of your protein and ligand. It directly has already implemented default settings for your MD simulations, and it can still be quite flexibly modified if you want to adjust some settings. Then the simulation part is used for preparing and running the MD simulation, and also post-processing the MD simulation so that it's directly then ready to be used for the analysis thereof. And for the analysis part, it consists out of different tools for the analysis, those being first the stable waters and the clustering of the waters, then the generation of the protein ligand interaction fingerprints, which allow us to create these binding modes, which are then the combination of interactions. And finally, then the different 2D and 3D visualization tools that we have in there to look into more detail between the protein and ligand interaction. And to quickly highlight the newest releases that I had, I uploaded last week or the week before that version 1.1, where I updated a couple of things, those being an implementation of Donatello, another tool that I created for the 2D visualization of the protein, of the ligand. Then we also implemented PyMol for an additional visualization option. Then for the ligand recognition, we previously had in OpenMMDL Analysis OpenBubble used for the ligand recognition if the user didn't specify it. Now we converted to a kit converter for it. And we then also added small things like the Smirnov force field for the PDB path so that you can select if you want for the force field for the ligand to be either Graph or Smirnov. And we added a Docker file because right now we are able to install OpenMDL with Conda install, but we also thought it would be nice to have the Docker file to also be able to install it to Docker. And the idea is then for the next release to basically have two important parts implemented, those being the addition of the post-transitional modification and the addition of prolif for protein ligand interaction recognition. Because currently the thing is that we in OpenMDL parameterize the protein separately and the ligand separately, so that currently it's not possible to have a covalent bound ligand in the MD simulation. And with the post-translational modification, it's possible to kind of do LD-kit chemical reaction to combine LD-kit to LD-kit the ligand with the protein to kind of covalently bind it with LD-kit, and then afterwards post-process it. Afterwards, do the force field generation so that it's possible to run an MD simulation in OpenMM of a covalent bound ligand with the protein in OpenMM. And the second part is then right now the implementation of prolif, because as previously mentioned, it didn't have water bridge interactions implemented at that time. So we use PLIP, but we would then now also include prolif as a second option for protein ligand interaction recognition, because it's quite often the case that PLIP and prolif have a little bit different algorithms of recognizing the different interactions. And sometimes the prolif are more reliable, sometimes PLIP is more reliable for the interactions. Correct, and to quickly wrap up my talk, I wasn't the only one coding and creating OpenMDL. I'm the main maintainer and main contributor to it, but there were also other people who helped with designing and creating OpenMDL, those being Nicholas, Yu, and Leon. Nicholas was then helping with different parts of the OpenMMDL analysis. Yu was mainly responsible for creating the Ember path in OpenMMDL Setup, and Leon was then the one who did mainly the coding for the water clustering to recognize the stable waters, so that he was the one who mainly coded for it. And obviously different people from the Volba lab, which I'm also a part of, contributed to these test cases so that we then had from each of the lab members a test case that we could then directly use to see if OpenMDL is used in their user test case. Correct. And I would say this is then it from my side from the talk. Do you have any questions for me?
Of course. Thank you.
Yeah.
Thank you, Varelji, for
Sure. You're welcome.
It's really an exciting talk, and
Thank you.
for someone who is passionate about using these computational tools, and even when it goes to the basic ones like PyMol, you start thinking in terms of envisioning these molecular substances in various dimensions. But for me, I think somewhere you presented or you talked about how prolif has been the way it has been designed to handle the fingerprints, and I think it is really novel. But then I think last year there was a paper about 3D, sort of like using the 3D sort of structure of molecules. I think using it to predict some sort of like binding affinity, and it got me thinking how sensitive is prolif, and I think the associated patterns in handling the orientations of orientational changes within the ligands, so to say, I would say.
So basically,
but also some sort of structure changes. Yes.
So basically for prolif, since I kind of then help co-develop it right now, from what I know is basically that for most of the recognition of the protein ligand interactions, what it does is it looks at the distance and the angle. So that basically it looks at the ligand, how it is in 3D with the protein, and it first looks at the distance between them, since basically for hydrogen bond acceptors and so on, there are specific distances that are then the cutoff for the interactions. And it first basically looks in 3D at the distance, how far it is from each other, the atoms of the ligand to the atom of the protein, and also what angle it is. And basically in the case of PROLIF, they are kind of then also encoded in PROLIF, that basically it says, okay, we look at that distance and that angle, and if it's in the threshold, then we kind of say that this is a interaction that we look at. And with PROLIF in theory is then also, since it's open source, possible to modify them if necessary, that basically if you want to say, okay, I think that this interaction threshold right now is 3.5, but I think it can be higher, like 3.6 or lower. To make it more restrictive, you can also modify it there so that you can then basically adjust in theory what you think are then the correct options for the hydrogen bond acceptors, the hydrogen bond donors, and hydrophobic interactions. So basically it relies on, and yeah, basically then the movement is then again here, depending during the movement, if it goes above or below the threshold, that it then recognizes this as an interaction or not. There was also a discussion during Google Summer of Code for one of the projects that it could be an interesting idea to have like soft cutoffs, in the sense that, for example, for hydrogen bond acceptors, you kind of have like this perfect distance and perfect angle. And I think other than that, it's a little bit worse. So there was also a potential idea for the future to maybe also kind of distinguish between the present interactions, in the sense that if you have a perfect angle and distance for a hydrogen bond acceptor, it's not the same as if you have a hydrogen bond acceptor interaction that is almost on the cutoff. If it moves a little bit, then it gets away. And this was then the idea maybe for next Google Summer of Code to have like soft encoded interaction recognition, that it's not ones and zeros, but sometimes something
Wow.
between it. Yeah.
You know, the more you talk about the work.
What? Can you repeat?
I think my talk is getting bad. I don't know. Can you hear me?
Yeah, I can hear you. Yeah, I think, yeah, I can hear you. Can you hear me?
Okay. Yes, I can hear you. Sorry,
Okay.
I think I interrupted you when you were speaking because I thought you had stopped.
Yeah. But yeah, basically,
but
Mhm.
yes, but based on what you were explaining, and I see now you giving the users all this power to determine
Yeah.
their requirements.
Yeah, correct. So
But
that they are,
and you talked about like how OpenMMDL is using like the ProLif, and you establish like determination of the angle and the distance.
Yeah.
And I'm wondering, from a computer science perspective, what sort of algorithms involved within this determination? But also, again, from someone who is a computer science enthusiast, and you care about performance, and I'm wondering, all this is built from on Python, like that's the underlying stack.
Correct.
Wow. Uh, it's an ingenious, but maybe you could give us an insight into, from a computational perspective, algorithm to determine, the distance, the angle.
Oof. So this can be a tricky one, because basically I know, since I'm not the one who coded like the recognition, I'm basically the one who then used afterwards the recognition. I basically then know more from the chemistry side, that how it's then defined, in the sense that, okay, we define this as hydrogen bond acceptors and this as hydrogen bond donors to the smart patterns, so that basically recognize them. And then for the coordinates, it's basically, since I assume, so basically since PROLIF is kind of connected to MD analysis, it basically then use the MD analysis package for recognizing the positions of the protein and ligand. So I assume basically it's somehow then, I'm not sure in the details of the code, but I would assume MD analysis goes in the direction of NumPy and so on to the coordinates somehow to get the coordinates and positions of the atoms. And basically then you kind of have like an array, something that direction. So I'm not that familiar in how in detail it works with the distance, what package it uses, but this would be the direction that I would assume, considering that NumPy is an part quite often used in MD analysis for different parts. So it would go in that direction. And yeah, basically from the speed and so on. So yeah, correct. It's OpenMDL is built upon Python, and basically the only option to increase it was, for example, for the different parts of to the analysis, which was then the main part that takes some time to recognize it. Here, basically, we thought to increase it, we just use multiprocessing and basically then start splitting up the processes to make it everything faster, to kind of then split up the processing of each of the frames separate on a separate CPU so that it increases the speed. In MD analysis itself, they didn't have it previously. Right now, they also implemented Dusk so that there's also a parallelization from different parts of MD analysis. So that basically this was then to kind of improve the speed of the calculation.
Okay. Wow. Interesting. In the slides somewhere, you emphasized the stable water clusters.
Yeah, correct.
And one, because I'm not so much a biologist, I'm more of a computer scientist, so software engineer. But I want to like, I'm wondering what's the focus much in performing simulations on trying to establish stable water clusters, and what role does these water molecules, like maybe would say, play, that we have to focus on them a lot to have these clusters identified? Yeah, so that's where my curiosity is.
Yeah, it's correct. So basically, we looked at it more from the chemistry side of things because quite often in proteins, like the alkene kinase example, there are quite often like interactions that are quite important to inhibit the protein that you need, like to have a specific water bridge interactions. For example, this alkene kinase, it had then in the literature that we looked at a specific, quite often interaction that appeared from time to time. And this was in the interaction that we have these residues and they have this specific water that we need to target to inhibit them. So basically that sometimes the stable waters are quite good in the sense that you can directly then use them as an additional interaction with the protein so that you have either the hydrogen bond acceptance and donors between the ligand and protein directly, or in some cases it is then quite important and good to have then these water molecules that you can then target in the sense that your ligand interacts with the water and the water interacts with the protein, and then you have an additional interaction with your ligand and protein that basically then has effect on the binding affinity of your ligand with the protein. This would be the first option, that you would basically target them. The alternative is that you try to replace it, because sometimes it's the case that you have these stable waters and they're kind of like trapped in the binding site, which is then sometimes good for the energy, sometimes not so good. So that sometimes it's also beneficial for inhibiting proteins or activating them to basically replace this part of the water with a part of the ligand. So for example, where you have a quite stable water, you instead put your, for example, you put their hydrogen bond acceptor or donor, like an amine, that does the same interaction with the protein, and then basically you kind of replace the position of the water molecule with a part of your ligand and get then the interaction there because it's quite stable, and this then basically improves usually your affinity, binding affinity of your ligand with the protein. This would be then kind of the second option, that you could then use the stable waters to know, okay, here like quite often waters interacting, and it's quite good to either use them for an additional interaction or replace them to have then an interaction there, because we know that these waters are then trapped in the binding site and this affects then the protein. So these are then the main focus reasons and points why we then created this algorithm. And this is then why we then used it for this ALK2 kinase example, because there we had then like a literature mention of those water bridge interactions being important for the compounds. And basically also in the example, we then saw that the more potent compound used it more and had then more additional interactions with the water molecule than the other one, than the less potent ligand. Yeah.
Wow. I see. And maybe my last question would be,
Sure.
MD simulations, like I feel, in my opinion, and I could be wrong, but I feel they are not democratized, and I think one of the reasons is one of the most of the tools that are involved are computationally expensive.
Yeah.
And I'm wondering, like, I would love to know about the computational sort of like needs of OpenMMDL, if
Yeah.
possible, even OpenMM. But
Yeah.
yeah, the computational needs mainly.
Correct. So basically here again depends on what hardware you have. In theory, basically OpenMM and then OpenMDL, because it's based on OpenMM, can you run MD simulations both on CPUs and GPUs. So that basically, if you have NVIDIA, you can use CUDA there to run MD simulations. Now they also have like HIP implemented from the AMD GPUs. So basically that in theory can use both GPUs and CPUs. Yes, it is basically for OpenMM kind of then prefer to have more GPUs. For the expensiveness depends on basically how much you want to run the MD simulations and how long it takes. So basically it's then quite variantly and different depending on what hardware you have, what GPUs you have, what CPUs you have. So basically in theory it's possible to run directly on CPUs. It will be slower than on the GPUs. Basically, and then again the GPUs depends on the graphic card. So for that one, I think for OpenMM they did a lot of benchmarking tests to see the performance of OpenMM in regards of using different GPUs and comparing it GPUs to CPUs. So here I don't have the exact numbers, but they did publish benchmarking papers for it. And basically again here it completely depends on what you have the access for, what hardware you can use. So here depends on what you can use.
Okay. At
Yeah,
least I get the impression that with laptop, I think I could have a chance on running OpenMMDL.
I mean that's possible. If I can ask, what hardware do you have so that I have like an estimation?
Yeah, I think I could be Ligand estimation, but also trying to maybe pick random proteins to see
Yeah.
what could possibly come out as a beginner to start using
Correct.
out and see
Yeah,
what could come up. Yeah. So
correct.
I want to thank you. I don't see any questions from the audience.
Sure. Happy to be here. And thank you very much for having me.
Yeah. And yeah, so I want to thank you. Thank you for
Yeah.
taking off your time and talk to us about this work. Yeah, so that increases the toolbox size when we want to perform
Yeah.
molecular dynamics. And I think this is a
Correct.
good intro.
Correct. I guess also what I could then quickly highlight, if you then, so basically because also another person that I know, another colleague, he does also something more in that direction of making it more user-friendly, but for docking. So basically that I can then also send you a link afterwards for a docking tool that basically also is then like OpenMDL, also open source, user-friendly, but in this case more docking direction. So that basically is then also quite
Definitely
interesting.
open. So
Yeah.
yes, definitely. Please, it
Sure.
will
That I
be.
can then.
I appreciate it
That I can
to
do, then I'll
explore
send
the
it.
tool.
Yeah,
Yes.
absolutely.
Yeah. So thank you very much,
Yeah,
Valery, and
You're
the
welcome.
audience.
hmm
And thank you very much. I can't thank you enough.
Yeah, sure. Glad
Uh,
to be here and glad to have presented my work here. Also interesting that there is a community in Africa that does a lot
yeah.
or is interested in computational drug discovery using AI. So it's kind of interesting because I'm quite familiar with the Europe field, basically of the research groups in
Yes.
Europe, but then less familiar with other regions. So it's then also quite interesting to see other regions and get more familiar with them.
Yeah, absolutely. Thank you very much. And I really, you can never, my thanks to the team that you worked with. Really appreciate accepting this invitation and the presentation altogether.
Glad to be here.
So I'll let you go. I will post this video on YouTube. Probably I'll tag you on Twitter.
Yeah,
and,
of course.
And yeah. So a goodbye to everyone, and a good evening to the guys in Uganda and those around the world. Someone is in South Africa and the US. Good morning, good evening, and bye-bye to everyone.
Bye-bye.
See you in the next sessions.